Hello everyone, I'm Marco Cristancho from Cafe, the National Coffee Research Center in Colombia, and I'm gonna tell you about our project exploring the soil-associated microbiome of Colombia coffee plantations, a bacterial and fungal taxonomic and functional analysis. The team that participated in this project was Valeria Fayori from INTA in Argentina, Lorena Gomez from the National Autonomous University of Mexico, Victor Hugo Ochoa, who is currently a PhD at the Arizona State University, and Maximo Rivarola, also from INTA in Argentina. Myself, at the time of the project, I was at the University of Los Andes in Bogota, but currently I'm at the National Coffee Research Center's Cafe. So we did sample 3 different sites in Colombia, each one representing very different coffee growing regions in the country, from the north, from the center, and from the south of Colombia. We sample organic and conventional farms as well for this study. The methodology, we did sample the 3 sites that I mentioned, 2 farms per site, and we did 5 replicates per every site sample. We perform sequencing by Illumina of the amplicons, the 16S amplicons, and the ITS regions for fungi. We also perform analysis of physical and chemical characteristics to try to correlate that with the species found in our analysis. So let's talk about the bacteria that we could identify by 16S sequence. From the bacterial communities, the phylum that we could identify, amplicos from all samples were assigned to 14 phyla, 24 classes, 14 orders, 23 families, and 42 bacterial genera. The diversity indices calculated from the rarefaction curves remain constant across the different treatments. Different bacterial communities prevail across the sample regions. The analysis of the beta diversity did not allow us to corroborate differences in the structure of the microbiota associated with the coffee soil in the different. Now for the fungi, we did the sequencing of the ITS region. Here is the analysis. The relative abundances of soil fungi show us that we could separate all samples by their unique fungi composition, as seen here at the level of families, and also here in this plot where we could see that all samples were separated but their fungi composition. This is a graph showing the most abundant genus of soil fungi that we could identify in our samples. The Saprotroph mortadella was broadly frequent and dominant. Cytosema was prevalent in the long-term agroforestry farms. And even though we could not assign an ecological function, these genes may hold a valuable potential to improve the beverage quality of coffee. The identification of beneficial fungi such as Metarhizium and Trichoderma, widely used as biological control agents in Colombia's coffee production, is an indication of the health of coffee soils in the country. In addition, no major pathogens of coffee roots, such as Rosellinia, were identified in any of the samples. It was possible to separate the species classified as saprotroph, symbiotroph, and pathotroph, and the combination of those species in the samples. And we could differentiate all the sites sampled by this identification of groups. So in conclusion, different fungal and bacterial communities prevail across the sample regions. This exploratory survey sheds light on the composition of soil fungi and bacteria in a poorly studied area, with the goal of providing important information for ensuring sustainable food production. The findings underscore the need for further research to examine how different management practices can enhance beneficial fungi and support sustainable coffee production in Colombian countries. So we would like to acknowledge the support of this project by the Cabana Grant on behalf of the Global Challenge Research and Cabana Net, who was funded by the Chuck Zuckerberg Initiative and who is providing funds for the participation of the group in the ISCB LATAM Solvio. We have two associated publications already for this work, and you can see them there, published in Tropical and Subtropical Agroecosystems and in Frontiers in Sustainable Food Systems. I would like to thank you for your attention.